That's it we're going to talk about this morning. Well, last time we talked about the structure, ATP, adenosine triphosphate. And I said you need to know that structure. And I wanted to talk a little bit about some things, but first, Haley, you have any announcements?
You might have seen in the group me, I've posted a couple questions the past 2 days of just a multiple choice. It's just a poll, so just treat it like a practice question, a test question. You can select an answer. It's anonymous, only you will see what you put. I don't even see it. So it's just to your benefit if you want to have some sort of daily practice. I'll be putting out those questions, so after this lecture I'll do one that relates to what we learned today. It kind of just gets you in the routine of making sure you're thinking about this knowledge every day, reviewing your notes, and kind of gives you a chance to check yourself. So take advantage of that if you'd like, as well as the review session Friday at noon.
All right. I want to speak a little bit to study habits, things that you can do to be successful, not just in this course, but as you move on in your college career. A lot of you, or some of you, I shouldn't say a lot, may use flashcards, and flashcards are really good for things like this, which is just rote memorization. But most of this course is not going to be aided by you using flashcards. You need to, just like Haley said, spend time with this material every day. You need to learn for understanding, not for just putting down exactly what the question is. It is so important to your future to be successful. Like I said, I've been on both sides of the coin. I've been on the side where I failed classes, and I've been on the side where I had a 3.98 at Texas A&M. I know what I'm talking about. I want to see you be successful. All right. So we're going to kind of continue our quick tour of organelles, of eukaryotic organelles. And this guy is the peroxisome. It contains an enzyme that's like one of the most efficient enzymes there is, called catalase. And catalase breaks down hydrogen peroxide into water and oxygen. And so the peroxisome is where you do a lot of different digestive things, detoxification, and they use these oxygen radicals, and it produces hydrogen peroxide. So it has this catalase in it to be able to get rid of the hydrogen peroxide. Where did I have it in my, I need to find out where I am in my notes. Oh, I don't need no stinking notes. I want to get to the side of the skeleton. All right. Let's just see what's next. All right, vesicles and vacuoles. So one of the things that is different between vesicles and vacuoles, vesicles can fuse with other membrane compartments or with the plasma membrane. Vacuoles cannot fuse with other membranes. They both are membrane bound. They're both involved in storage of things. The central vacuole of plants, we'll see, is a very important. Vesicles are used for transport within the cell. So we talked about things branching off from the Golgi, these vesicles that branch off from, the ER and are transported to the Golgi, and from the Golgi to wherever. Well, those vesicles are being used as transport vesicles. All right. So you can transport things from organelle to organelle with vesicles and vacuoles. One of the key aspects of what you'll be asked to do is, I said one of our learning objectives was to be able to compare and contrast animal cells and plant cells. So I made this 2 slides to go quickly through the things that are the same and different. All eukaryotic cells have a nucleus and a nucleolus. Prokaryotes don't. We're talking about eukaryotes here, plants and animals. All of us plants and animals have the endoplasmic reticulum, which is continuous with the nuclear envelope. We talked about that. You got to have ribosomes. Even the mitochondria and the chloroplast have their own ribosomes. Okay. Plants and animals have the Golgi. Well, it seems pretty easy so far. They share everything, right? Yeah, they both have mitochondria. Plants don't make sugars for you, they make sugars for themselves. The mitochondria help them harvest that energy back that they put into those sugars. All cells have a plasma membrane. Both plant and animal cells have microtubule organizing centers. However, within the MTOCs, only animals have centrioles and centrosomes. So it's kind of a subpart of the MTOC, but only animals have those. The peroxisome, what we just talked about, yeah, you find that in plants and animals. They have the same reactions to deal with. The cytoskeleton, which we're going to talk about in a little bit more detail today, yeah, they both have cytoskeletal elements. These are proteins that give the cell structure, that can enable the cell to move. So, yeah, they have. So on this first page, only have one thing that's not checked on both. The next page is going to be a little different. Chloroplasts are plants only. It'd be kind of cool if we could put chloroplasts in ourselves, and you could just go outside and breathe some carbon dioxide, and you would be getting all your food. I mean, not all your nutrients, but some food. Other plastids are also plant organelles. We typically talk about chloroplasts, but there are other plastids. So we just kind of have this general category out there. Lysosomes are animal only. We're going to talk more about different types of cell junctions and things, but plasmodesmata are these connections between cells and plants. And when plants make a connection between cells, that connection has to go through the cell wall because plants have cell walls. So these plasmodesmata are connections between cells, and there are these channels in the cell walls, if you will, that allow these cells to connect. Otherwise, they would be completely separate. Cell wall, that's found in plants and fungi, but we're talking plants and animals, so we're going to leave that as plants. And the central vacuole is a structure that is plant only. So when I look at this, it looks like plants have a lot more things that they do beyond what we do. Otherwise, we pretty much share the same things. Right? So maybe plants are more evolved than we are. All right. The centrosome. I mentioned that the centrosome is found in animals, not in plants, but that both had these things that we call microtubule organizing centers. Microtubules are part of the cytoskeleton that we're going to talk about in a bit. They're also the highways for the cell. We'll talk about that. But one of the main things you need these microtubule organizing centers for, maybe not the main, but the one that's most apparent is during cell division. Microtubules come from the MTOCs and go to the chromosomes, and we call them spindle fibers, and those are the things that are responsible for pulling the chromosomes apart during cell division. Okay, so the centriole is actually made out of some proteins, well, all proteins actually, but it has microtubules, and it's a 9 plus 2, oh, I don't need to know that. It's an arrangement of microtubules. You have these two short sections of microtubule structure called centrioles, and they're at 90 degrees opposed to each other. Plants have cell walls. Now there are, bacteria have cell walls with peptidoglycan. Some fungi have cell walls, but since we're talking about plants and animals, we want to talk about the cell wall in plants. It's mainly cellulose. A lot of you are wearing cellulose in the form of cotton. A lot of you are writing on cellulose in the form of paper. All right, so what is cellulose? Well, you're writing on sugar and you're wearing sugar. You're wearing glucose and you're writing on glucose. Because cellulose is just one glucose attached to the next glucose, attached to the next glucose, all in a line. What makes it different than starches, which are one glucose, chain to another glucose, chain to another glucose, is the kind of bond it has. It's still a glycosidic linkage, but the conformation, the geometry of the bond is different. So we call it a beta 1,4 glycosidic linkage. If you remember, with starches, it was an alpha 1,4 glycosidic linkage. So this little tiny change in the geometry makes this huge change. Starches are kind of have this curve to them and they have branches to them. Cellulose is just a straight line. And it's because of this beta 1,4 linkages, and there's no branches in cellulose. The paper fibers are a bunch of little fibers of cellulose that are all just all tangled together. They're bound by being tangled up. Don't have branches going between each other. Okay. The chloroplast is a organelle that's very much like the mitochondrion. I talked about the theory of endosymbiosis. The idea that these early cyanobacteria or early photosynthetic cells were engulfed by precursors of plant cells, by cells that already had mitochondria. And from those cells that engulf these guys, we have the line of all the plants you know. And so it has a couple key things. Now, I did modify this from your book a little bit. So I put in circular chromosome and ribosomes because those are important. It has its own DNA, just like the mitochondria, its own circular chromosome, just like the mitochondrion, just like bacteria. It has its own ribosomes, and those ribosomes, like the mitochondrial ribosomes, are more closely related to bacterial ribosomes than the ribosomes just outside of it in the rest of the cell. All right. It's a triple membrane system. So we have this compartmentalization. We'll start with the outer membrane. That's pretty obvious. But then there's an inner membrane, and that means we have this inner membrane space. And every time we have compartmentalization, that means we can have a different chemistry, a different set of proteins and fats and carbohydrates that reside in those compartments. So that also gives us this space in the middle, which we're going to call the chloroplast's matrix, or stroma, is what I want to say. Matrix is mitochondria. I'm getting my mitochondria and chloroplast confused in my head right now. Matrix is mitochondria. Stroma is chloroplasts. So all of this kind of yellow stuff, this thing that we would call in a cell, it would be called the cytoplasm. In the mitochondria, it's called the matrix. In the chloroplast, it's called the stroma. Then we have our third membrane system. And these are hollow, interconnected things that we call thylakoids. Each one of these guys is a thylakoid, and this one's been cut in half so that you can see that it has its own inner compartment, so we have even more compartmentalization. And when we stack these thylakoids together, we call the whole stack a granum. So a granum is just a stack of thylakoids. But you'll notice these corridors are are these interconnections. It's all an interconnected system, much like the nuclear envelope and the ER. So I already talked about endosymbiotic theory. I found this, I forgot to put the publication in here, from a different publication. So I wanted to just share this with you. So here we have our proto-eukaryote. In other words, it's not a eukaryote yet, still more of a prokaryote, but it's on the way. And what happens is we think there was some vagination and it starts making its own things like the endoplasmic reticulum, and then it engulfs an aerobic bacterium. That is what becomes the mitochondrion. And then this guy, some of these guys, or at least one of these guys, probably some, started engulfing these photosynthetic bacterium. And in both cases, they didn't digest the bacterium, they kept it, and the bacterium became a symbiont. It produced things that the cell needed, that the cell could use. In the case of the mitochondria, it took pyruvate and started dumping out tons of ATP. Now this cell had so much energy that it could move anywhere it needed. It could do all sorts of new cool things. And in the case of the chloroplast, this thing's making food out of thin air. Carbon dioxide, water, sunlight, food. It's pretty amazing when you think about it. All right. Any questions on that so far? So the central vacuole, like I said, is something, it's a structure found in plants. Now plants have cell walls. Have you done the osmosis in your lab yet? Okay, so you've talked about tonicity a little bit. Good, because that's what we're going to talk about now a little bit. So the central vacuole, plants are kind of weird. They actually prefer, to some extent, a hypotonic condition. That is, they want an influx of water because they use that pressure of the water, that osmotic pressure, to push out on the cell wall, and that makes the plant stand up. In an isotonic condition, the plant will still stand up, but it won't be quite as rigid. And in a hypertonic condition, the plant is wilted because you can see the central vacuole collapses to this little tiny thing, and the cytoplasm around it all gets sucked in, and almost the water's gone into that's left has gone into the vacuole, and the water that's gone out of the cell, so that plant's not doing really well. Okay. So this idea of tonicity directly has effect on how plants act. It also has effect on you. If you ever go to the emergency room and you get just general fluids, they're probably going to give you normal saline. Saline just means salt. Normal saline. They don't put water in your veins. That would be bad because of tonicity. Normal saline has 0.9% salt, which matches the tonicity of your blood. Okay. So this idea, the same things will happen to your cells. If we put water in there, they don't have a cell wall to contain things. So if I put water in your veins, your red blood cells would expand until they burst. It wouldn't be contained by that cell wall because they don't have the cell wall. All right. All right. The endomembrane system. We've talked about these different structures before: the nucleus, the rough ER, the smooth ER somewhere, the Golgi apparatus, transport vesicles. But they work together as a complete system. Proteins that are membrane bound, or proteins that are bound for secretion, the ribosomes make into the lumen of the ER. The smooth ER makes lipids. That is, like more things like plasma membrane, that's lipids. These vesicles pinch off from the ER, and then they are transported to the cis face of the Golgi. The cis face of the Golgi points toward the nucleus. closest space of the Golgi to the nucleus. The Golgi processes those fats, carbohydrates, proteins, and it modifies them and packages them and sorts them. So what happens is the proteins and things all come in here at the cis face, and then what this doesn't show are the little vesicles that bud off and come here, or sometimes this whole thing becomes the next layer. You'll learn that more in cell biology. But the progression is from the cis face to the trans face. The trans face is the face that faces the plasma membrane. As it goes through the Golgi, the protein or lipid is getting more modifications, carbohydrates put on it, and they're also getting sorted so that they wind up on the right vesicles. So there's some vesicles that are going to be involved in exocytosis. This one is actually not exocytosis. This is showing a membrane-bound protein with probably a carbohydrate there, and this vesicle fuses, it gives more plasma membrane, and you can see our protein with the carbohydrate is now in the plasma membrane. That's how we get these integral proteins into the membrane. They're actually made into the membrane here and packaged and sorted in the Golgi. Ah, there's my smoothie R. I forgot. I knew it was somewhere. The smoothie R is also continuous with the rough ER, which is continuous with the outer and inner space of the nucleus, the nuclear envelope. All right. The sarcoplasmic reticulum is a specific kind of endoplasmic reticulum with a specific purpose. We find the sarcoplasmic reticulum in your muscle cells. And the main function of the sarcoplasmic reticulum is to be a warehouse or storage center for calcium ions. When you want to move your finger or your lips or your eye, you need to contract muscles. The signal to contract is a release of calcium ions. So those ions are stored in the sarcoplasmic reticulum. They go out and cause contraction, and then they're pumped back into the sarcoplasmic reticulum until the next contraction is needed. So this is a muscle fiber under all of this, and you can see the structure of the sarcoplasmic reticulum just kind of wraps these fibers so that they can release calcium, and it gets released over the entire cell contractile unit, and it can take up that calcium quickly. Meaning I can move my finger, my lips, and my eyes, and all of those things. I'm not worried that you know all of the triad and T-tubule at this point. But I do want you to know this thing that's called the sarcoplasmic reticulum is a form of ER that we find in muscle cells. And this is a better shot of the Golgi. So you can see from the Golgi, we get lysosomes. That's where they come from. And we get transport vesicles to go to other places. And we get this, here's a protein for secretion. This would be exocytosis. This guy fuses with the cell membrane. These guys are free to go into the extracellular space now. So that's just showing how all of this system works in concert as a complete system for packaging proteins and lipids, modifying them with carbohydrates and other modifications. All right. Finally, getting to the cytoskeleton. In the cytoskeleton, we have a couple different types of fibers. I'm going to draw your attention to this one that's on the periphery first, called microfilaments. That always throws me off because I never call them microfilaments, because the other name for them are actin filaments. The protein they're made out of is called actin. Actin is also found in your muscles. It's a primary part of that contractile unit I was just talking about. So these actin filaments are involved in cell movement. So when you see cells all kind of crawling around like the bacterium we're going to see getting chased by a white blood cell, a lymphocyte. It's by the restructuring of these actin filaments. All right. The next thing up are these intermediate filaments. And intermediate filaments have a little bit different structure than the other filaments. They're basically structural. But things that you've heard of, like keratin and collagen, are different types of intermediate filaments. So your skin, you need collagen and keratin and all of those good things to give your skin the structure that it needs. Those are intermediate filaments. Finally, this would be a cell in the middle of division because it has two MTOCs. So this cell must be dividing. So there's the metaphase plate right there and all sorts of chromosomes getting pulled apart, but they didn't draw that. But just by that diagram, I can tell that cell is dividing. But we have these microtubules. During cell division, they act to pull the chromosomes apart and help push the cells apart to form two daughter cells. During normal cell activities, which we'll call gap 0, there's only one MTOC. The microtubules are still there, but now they act as highways for transporting those vesicles to where they need to go. There's actually little motor proteins that hang onto the vesicle and walk down the microtubule. It's really kind of cool. To their destination. All right. So here we go. Microfilaments are actin filaments. So actin filaments are made out of these globular or spherical subunits, but a whole bunch of them get chained together to make filaments. And so that's true of whether we're talking about at the periphery of the cell or we're talking about muscle cells, whether we're talking about cardiac muscle or smooth muscle. This is, I don't expect you to recognize all of this stuff right now, but this is a really, really close up of part of a muscle, of how the muscle actually works, part of a contractile unit. And what happens is we have these motor proteins that we call myosin. And these myosins have a tail, and all the tails get bundled together, so this is all the yellow-green there is all myosin. But myosin have these heads, and they basically attach to the actin, and they push the actin. Then they reset, and they push the actin. So when I'm doing this, I actually have all sorts of myosin motors that are pulling on the actin to make my muscles contract. And this was kind of actually an interesting paper, but you guys don't care about that right now. All right, so another thing that actin is involved in is the, like I said, cell movement. And I want to, hopefully this works. Let's see if it's going to work today. So this is how I learned, this is what I learned that white blood cells look like. With modern technology, this is what white blood cells look like. All of those surface changes you see are the remodeling of the actin part of the cytoskeleton. Okay. So these actin filaments are super important for this little guy to be able to chase them. Looks a lot different in 3D than it does in 2D. I just wanted to play the music. All right. I want that. Where's the next slide? Next slide. There we go. All right, so we had our actin or microfilaments. These are the narrowest filaments in the whole system of your cytoskeleton. Then we have, I talked about these intermediate filaments. And these are long, instead of being globular like actin, they're these long proteins, and they're just kind of twisted around each other like you might twist a yarn together. And then they have these interactions. This is trying to show those non-covalent interactions that keep them bound together. Sometimes the interactions are covalent, like disulfide bonds. But this is keratins, desmin, vimentin, neurofilaments, lamins. So intermediate filaments are a little bit different than actin and microtubules. In actin, we have the actin subunit, and we make actin. In microtubules, you'll see that we have alpha and beta tubulin, a specific protein. Intermediate filaments actually refers to a whole host of different proteins that have the same basic kind of structure, but have some different functions in different parts of the body. All right, my favorite, microtubules. My buddy in grad school, we had a lecturer who was actually a world famous guy, but we made fun of him because he called them mycotubules. So we used to call them all mycotubules. Microtubules. These are hollow structures, again, made out of a globular protein or globular proteins. There are two subunits to actin. There's alpha actin and beta actin. And they come together to form these structures where we have 13 dimers. That means a dimer is, we have two things. So we have a beta actin and alpha actin, or I should say alpha and beta. That's a dimer. And there's 13 of those dimers that form these rings. And these rings come together to form these tubules. Now, we're not going to talk about treadmilling and all sorts of really cool stuff right now. You'll learn that later. But I do want you to know that microtubules have a polarity. They have one end that we call the minus end and one end that we call the plus end. All right. That brings us to this idea of the extracellular matrix. So far, we've been talking about the cytoskeletal elements that are inside the cell. But cells excrete things like the intermediate filaments like collagen. They have fibronectin that hooks up to the collagen. They have all of these carbohydrates, all of these little tree structures are different carbohydrates. Here's the protein and here's the polysaccharides. Polysaccharide and carbohydrates are kind of the different names for the same thing. Right? But I took this from your book, so we'll just go with it. So they have these proteins that are decorated with all these tons of different carbohydrates. And those carbohydrates act as signals to say if that cell belongs. Your antibodies, when you get invaded by a bacteria, respond to lipopolysaccharide. You don't make lipopolysaccharide. That's one of the things they respond to. But bacteria display these lipo, which means fat, poly, many, saccharide, sugars, carbohydrates. That's one of the key things that tells your body this cell does not belong. So we have this thing that we call the extracellular matrix. Notice down here we have our part of our cytoskeleton, the actin filaments, or as it shows here, microfilaments. And then we have these transmembrane proteins that we call integrins that tie both the microfilaments, i.e. actin cytoskeleton, to the extracellular matrix, helping anchor the cell in place. All right, intracellular junctions. We're going to talk about a couple types of ways cells communicate with each other directly. And I already talked about plasmodesmata, these connections between plant cells. Animal cells also have these intracellular junctions, but we don't have cell walls, so we don't call them plasmodesmata. What you see here is the arrow is trying to show you where we have these connections between cells. It's not necessarily all that apparent there. Trying to figure out what I'm looking at. Let me see, I see all the nuclei. I have some idea what that might be, but I'm not sure. Okay. So let's talk about the plasmodesmata. Here's our cell wall, and the cell wall has these gap in it, and that allows the cytoplasm from the two cells to join. So they're two independent cells, but they are in direct communication. Molecules can diffuse from either cytoplasm to the other cytoplasm through these plasmodesmata. And the only thing I don't like about this diagram is it shows pathways to the cytoplasm, and yeah, but also it can go, the arrows can go the other way. Depends on a bunch of different conditions. I understand what they're trying to say. In animals, your cells, we have what we call gap junctions. In gap junctions, we have our plasma membrane of the two cells. So this is the plasma membrane of one cell. This is the plasma membrane of another cell. Right between these two guys is our extracellular space outside of the cells. And this protein, this group of protein called connexins, can open or close. When they open, the cells, just like in plant cells, are in direct communication, cytoplasm to cytoplasm. When they're closed, the cells are not in direct communication. Okay. So they typically form these kind of little things like this, where you have a whole bunch of connexins, and we have this gap junction channel, and the connexins can then open or close. We also have things that we call tight junctions. In your intestine, for instance, it's really important that the food you eat gets digested and absorbed as nutrients, but you don't want all that stuff just going in willy-nilly wherever it wants in your body. You want to contain the stuff in your digestive system, like all the microbiota you have living in your digestive system, inside the digestive system. So cells here and in other parts of the body have these things that we call tight junctions. The cells are not in communication with each other, but they are tightly bound to each other, so tightly water can't get through. That's how you make a watertight vesicle. Well, the cells, tight junctions. I should say vessel. So this is just showing the intracellular space, and you can see these transmembrane proteins and strands, and they are the things that form these tight junctions. Desmosomes are kind of like tight junctions, but not quite as tight. I kind of found it interesting. Your book uses the term like a spot weld, which is, I'll take that. They have a fairly complex structure. I'm not worried that you know the structure, but where we might have a tight junction up top, these are not necessarily, these desmosomes aren't necessarily for keeping things watertight, but they're for keeping things hooked together. A spot weld. All right, any questions on, I can't look at that clock, can I? Okay. Any questions on what I covered so far on the whirlwind tour through the cell organelles? Because now we're not getting out of organelles, we're going to go in deeper detail of some of them, starting with the plasma membrane. This is chapter 5. The structure and function of the plasma membrane. So where we were talking about, okay, here's the plasma membrane, his phospholipids, we're going to get in a little bit more detail now. And I put this slide in because I think there's some question, well, why do I have to learn this Bio 101 or 1013 stuff? I just want to learn the fun stuff. I want to be a physician. Something as simple as the plasma membrane is a disease center. There are diseases of the plasma membrane. If you're a physician, you have to know and understand the diseases of the plasma membrane, how the plasma membrane works, so that you can understand the problem with the disease processes. Plasma membrane integrity can affect skeletal muscle, a contraction, and chronic myopathies. Myo means muscle, pathes means disease. So you have these chronic means long-term muscle disease. Your heart is cardiac muscle, and it can also suffer contraction issues and acute injuries from plasma membrane diseases. Immune cells can trigger pore-mediated injuries. Neuronal membranes can get injuries. And there are migration injuries that occur to immune cells, to fibroblasts. Fibroblasts are your skin cells that make fibers, basically all that extracellular matrix and things. That's why they're called fibroblasts. Blasts things, they make things. Osteoblasts make bone. Fibroblasts make fibrous extracellular matrix. And cancer cells. Of course, in the case of a cancer cell getting injured, we're kind of like, yay, but it all comes together. You want to understand cancer and how you can fight cancer? Well, this fundamental knowledge is what you need to be able to take yourself to where you want to go. So why am I torturing you with all this stuff like the structure of ATP? Because it matters. Because you have stated that you want to be a biology major. Everybody in this room is a biology major. There's, of course, 4 majors. I want you to be excited and love biology as much as I do. But you have to build the fundamental framework, and that's what we're working on. Okay. So, any questions on any of that so far? Got to check my time again. I got 5 minutes. I'll talk a little bit about this idea of the fluid mosaic model. So, when we talk about phospholipids, we drew a phospholipid, something like this. I had an R group of some kind, and that's going to be I'm going to put polar. Going to have a charge, and then I'm going to have my phosphate down here. And then this is going to hook up to my glycerol. So I'm going to make this an easy way to do this like this. I have my oxygen here, and this is going to be, we can just do it like this. Oh, no, we can't do that. Let's just do it this way. I'll put the first carbon here. There's a double bond oxygen here, a double bond oxygen there. Zig, 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 zig. A saturated and unsaturated fatty acid is what I drew there. So this is the basic structure of a phospholipid. We have a chemical group here. There's a whole range of them. There's phosphatidylcholine. Why am I not thinking of other things besides phosphatidylcholine right now? That's bad. In any case, there are these polar molecules hooked up to a phosphate group that's hooked up to a glycerol molecule that's hooked up to 2 fatty acids. The fatty acids are hydrophobic. They don't want anything to do with water. Water doesn't want anything to do with them. Up here, the phosphate group has a negative charge, and you got a polar group up here, which means water's gonna love it. It's got a charge. Water loves things with charge. Water's polar. So we say this is hydrophilic, and this is hydrophobic. And so that structure gives us this thing we call a lipid bilayer, where we have all of these phospholipids this way, and they self-assemble. What I want to get across is some of those phospholipids are bound to proteins that are bound to cytoskeletal elements. But for the most part, there's nothing holding this phospholipid to the phospholipid next to it, other than the fact that they're in an aqueous environment and it's energetically unfavorable to expose the fats to water. So these things are very flexible. These lipids, these phospholipids, can float around, they can spin around, they can move all over. They're not stationary by any means. So we have this fluid mosaic model. The idea that we have this very fluid plasmid membrane. We can change the viscosity of it by adding in cholesterol. We can also change the viscosity of it by using certain preponderance of certain R groups. We have lipids that have carbohydrates. Notice the carbohydrate-studded lipids and the carbohydrate-studded proteins, i.e. glycoproteins and glycolipids. Those carbohydrates point out toward the extracellular matrix. We saw there's all sorts of carbohydrates out that way. You don't see any carbohydrates on the cellular side of the membrane, but here are our actin filaments. We have proteins that are integral to the membrane. We have peripheral proteins. And this one I really did for the, no, I didn't do this for the music, but it's got some strange music. Baby? You can see the proteins and the lipids are flexible, they're free to migrate. You can see they're not connected. That's why it's called a fluid mosaic model. All right. So the mosaic comes from the fact that it's studded with proteins. In fact, by mass, over 50% of the mass of your plasma membranes are proteins. Now, proteins are really big, so they have a lot of mass, but nonetheless, over 50% of the plasma membrane is proteins, and proteins give the plasma membrane function. And I need to shut up now. I'll talk more about the plasma membrane next time. Any questions? All right. Today wasn't too torturous, was it?